Hi, everyone. I'm Samantha Nyer. I'm a PhD candidate at the School of Marine and Atmospheric Sciences, where the past few years I've had the privilege of working with the Center for Clean Water Technology. And today I'm going to talk to you a little bit about the work that I've been doing on evaluating nitrogen transformations in constructed wetlands for wastewater treatment. Before I get started, I just wanted to take a moment to let everybody know that the center is participating in Stony Brook University's Giving Day, which is on October 8th this year. Essentially, Giving Day is a 24-hour fundraising event. Funds raised from Giving Day will go directly to support our research, which is aimed at developing novel and affordable approaches for nutrient and contaminant removal from Long Island waters. On this presentation, I provided a link on the slide. I'm going to also post this link to the chat for anyone who's interested in learning more information about Giving Day. But generally, we really appreciate any support, either through direct donations or just by helping spread the word about Giving Day and about the work that the center is doing. And so today I'm going to talk about one particular environmental contaminant and one innovative approach to the nitrogen problem on Long Island. So many of you probably know that nitrogen is an essential nutrient, but we have overloaded our environment with nitrogen due to the rise in population, human population, and advances in technology, which have made it easier to produce forms of reactive nitrogen. And this has had a number of detrimental effects on human health, economic productivity, and the quality of our ambient freshwater resources and ecosystems. Most of the population, over 60% worldwide, are connected to, So in order to first address this nitrogen problem, we need to understand where the nitrogen is coming from. And so many studies have been done to try and clarify the sources of nitrogen. This is one such study that shows the nitrogen loading to the subwatersheds of the North Shore of Long Island. While the sources of nitrogen do vary geographically depending on where you are, what you'll notice in this figure is that a lot of the nitrogen actually comes from wastewater, which is shown in these blue and purple colors on this figure, whether it be a municipal sewage treatment plant or on-site systems. Globally, most of the population are connected to centralized wastewater treatment plants. However, these forms of treatment are not always economically the most viable. And so there's been an increasing trend in decentralized systems, which are used to treat individual or small groups of properties. And these types of systems are more cost effective than traditional activated sludge systems in some cases. And they can also be used to recover nutrients and energy, which is an interesting application since globally we're facing water scarcity. So the fact that we can use these systems to recover some of that water and some of those nutrients for example, agricultural purposes, is a really interesting potential application for these systems. And one technology that we're really interested in exploring a little bit more that's being applied as an IA system, innovative alternative on-site wastewater treatment system, are constructed wetlands. Way back in the day, natural wetlands were used as dumping grounds for waste. And during that time, it was discovered that wetlands have the ability to purify water. So they have kind of become analogous to nature's kidneys because they do such a good job at removing waste products. Constructed wetlands are systems that are mechanically simple, but biologically quite complex. They're engineered systems that are designed and constructed to utilize naturally occurring processes, but they do so within a very controlled environment. And this environment is more resilient and robust against shock loading and changes in conditions. Constructed wetlands can be operated in various configurations. They're typically comprised of a gravel or sandy media, and they rely on plants and microbes to treat pollutants from water through a combination of naturally occurring chemical, biological, and physical processes. In addition, these systems are attractive because they are relatively passive, low maintenance designs, meaning that there's not very many moving parts to them. We can carefully engineer these systems to target contaminants that we're specifically interested in treating. And in the case of wetlands, they offer aesthetically appealing designs, where above the surface, it can appear to be just a pretty garden on your lawn, but below the surface, it's actually serving a really critical functional purpose. And so within these types of systems, nitrogen is treated through typical microbially mediated processes of nitrification and denitrification, whereby we convert reactive forms of nitrogen into unreactive forms such as N2 gas, which is the majority of our atmosphere. It's what we breathe. And so in the case of treatment from wastewater, ammonium, which is the main form of nitrogen in septic systems, gets converted to nitrate in the process of nitrification under high oxygen conditions. But we don't want to just stop at nitrate. We want to further reduce this nitrate all the way to nitrogen gas in the process of denitrification in the presence of low oxygen conditions. Unfortunately, along these treatment pathways, there are a number of potentially harmful byproducts that can form. One example of that is nitrous oxide, which is a greenhouse gas. It has a very high global warming potential, and it can last a very long time in our atmosphere. So when we're designing these types of wastewater treatment systems, we want to make sure that we're not simply swapping one form of nitrogen pollution for another. In general, these nitrogen cycling in these systems require a variation of oxygen concentrations. And so, as a result, environments that generate a close juxtaposition of oxygen-rich and oxygen-depleted conditions strongly influence nitrogen removal. In constructed wetlands, an integral part of treatment are plants. And wetland plants do create unique environments where these processes can occur in several ways. Here I'm showing an idealized perspective of what can happen in a planted constructed wetland. I'm highlighting two distinct regions here. The black box indicates what we are going to call the bulk sediment, which is a region of soil that is not influenced by plant roots. In this black dashed line, I'm indicating a region of soil that is directly influenced by plant roots, which we're going to call the rhizosphere. Wetland plants are kind of unique in that they have to develop adaptations that allow them to survive in harsh environments. And one such adaptation that they've developed are these low resistance pathways for gases that allows them to transport oxygen from above ground to the soil. This creates a region around the root that's oxygen rich. And this size of this oxygenated region can vary. Plants are also known to release 40 to 60% of the carbon that they fix in the form of root exudates, which are labile sources of carbon that provide a really easy and attractive food source for microbes. And so in this way, plants create a very favorable habitat that support microbial communities. Here I'm showing a little bit of an idealized schematic that shows the relative distribution and abundance of microbes shown by these little Pac-Man-like creatures, in the root zone and in the bulk sediment. A lot of studies have shown that the abundance and activity of microbes is significantly higher in the rhizosphere compared to the rest of the soil. Since the rhizosphere provides the proper environmental conditions required for nitrification and denitrification, and since it supports a microbial community that carries out these processes, we believe that the root zone is a critical region for nitrogen removal to occur. So here I'm going to show what may happen in a wetland that's used for wastewater treatment as the nitrogen moves through the system. First, we have ammonium that comes in with the wastewater. This is, again, the main form of nitrogen in our waste. As it moves through the system, it encounters a plant root zone, which is oxygen rich. This can allow for the conversion of ammonium to nitrate in the process of nitrification. Once this nitrate moves away from this oxidized region, it can subsequently become denitrified to N2 gas, provided that sufficient carbon is available. And then this nitrogen gas can subsequently leave the system. So these are some of the processes that we think are occurring, and I want you to kind of keep these things in mind as I go through the data from the systems that we've been working with. With respect to the center's wetland initiatives, I want to highlight a few recent developments. First, we've recently partnered with the Nature Conservancy in the construction of a wetland at Uplands Farm. This system is a novel two-stage system that aims to promote nitrification and denitrification. And additionally, these were planted with different assemblages so that we can test the effects of plant communities on nitrogen transformations and nitrogen removal in the system. Unfortunately, modifications to the system were finished in March or April actually of this year, which coincided with the onset of COVID. And so as a result, we have not been able to sample the system yet, as it's currently not receiving any flow. But we are really excited for the opportunity to begin to sample the system and to see how it's functioning. In addition, the center has worked to create a constructed wetlands guidance document, which is intended to provide guidelines for the design, installation, operation, and maintenance of wetland systems on Long Island. This document is really geared towards engineers, installers, and maintenance providers. And much of the work that I'm going to show you today really went into informing the development of this guidance document. So the first field installation that I want to, the first system that I want to talk about is the first constructed wetland of its kind to be installed in Suffolk County at Sylvester Manor on Shelter Island. This was designed by Natural Systems Utilities. The system is unique in and of itself because it's a seasonally operated system. It's meant to treat restrooms on site that are used only in the spring and summer months. And so data from the system can inform designs for example, seasonally occupied households. So this is a very interesting system that we wanted to look at. The design of the system itself is also a bit unique. This is a schematic of the recirculating gravel filter. The top 36 inches of the system is made of pea gravel, and it functions as a conventional aerobic filter. So this is a very oxygen-rich environment where nitrification can occur. Below this are 18 inches of drain-field rock that's enclosed in a liner, a plastic liner. And so this creates a saturated submerged zone where oxygen is not very abundant and where denitrification can occur. So here we have a vertically stratified system that can promote these nitrogen removal processes. The system, just a few more details about the system. It's 420 square feet, designed to receive a flow of over 1,800 gallons per day. To give you just some sense of the sizing of this, an individual family household is expected to produce around 400 gallons per day. So this system is really the equivalent of, say, 4 to 5 households. It's a recirculating gravel filter, which means that wastewater makes several passes through the filter before it's released to a leaching field. This is different from a single pass filter, as it allows for increased contact with plants and media to enhance treatment. Additionally, because it's a recirculating filter, we are able to load it at higher rates, up to 5 gallons per square foot per day. And so because we can increase the loading rate, we can decrease or reduce the footprint, which equates to reducing costs. What I'm showing here are the performance data for the entire operational period of the system, excluding 2020, because we were not able to sample this season due to restrictions related to COVID. So here I'm showing effluent concentrations over the 3 sampling periods, and I've divided everything by month. The bars themselves contain a lot of information about the various nitrogen species present in the effluent. The white indicates nitrate, the black bars indicate ammonium, and the gray is the organic nitrogen component. And what you'll notice is that over the entire operational period, the system achieved average effluent concentrations of less than 10 milligrams per liter. And this is really significant because Suffolk County has set limits on effluent to be 19 milligrams per liter, and the center is targeting technologies that can achieve effluent concentrations of 10 milligrams per liter or less. And so this system meets both of those standards. The main form of nitrogen in the effluent is nitrate. And this is a good thing because it shows us that the systems are nitrifying. But also we can do things and implement things to further improve the nitrate removal in the system. So one thing that we know limits nitrate removal in systems is the amount of available carbon. This figure represents data from pilot systems installed across Long Island, and it shows the typical influent composition. The black solid line represents total nitrogen concentration. The gray line with square boxes represents the dissolved organic carbon concentrations. These concentrations correspond to the left axes. The white bars represent the ratio of carbon to nitrogen in the influent, and these values correspond to the right axes. On Long Island, average nitrogen in the influent is around 100 milligrams per liter. DOC, dissolved organic carbon, is around 67 milligrams per liter, and the average ratio of carbon to nitrogen is less than 1. This is kind of interesting because studies have shown that by increasing the carbon to nitrogen ratio in the influent, nitrate removal could be substantially improved. This is one such study that sought to examine how nitrogen removal is improved with increasing C to Ns. And what you'll notice is at a C to N ratio of 1, which is kind of in line with what we're experiencing, a nitrate removal is significantly impaired to what it could be at higher carbon to nitrogen ratios, even ranging between 2 and 5. So while we can't change the composition of the influent, there are things that we can do to try and overcome this carbon limitation. And so we installed a system to try and overcome this carbon limitation by adding an external carbon source. We did this at the Massachusetts Alternative Septic System Test Center in Falmouth, Massachusetts. Here we set up a design that was meant to test a two-stage system that consisted of nitrifying recirculating gravel filters and denitrifying carbon-rich media barrels. The goal of this project was twofold. First, we wanted to just provide proof of concept for this design. And secondly, we wanted to assess the role of plants in nitrogen removal. In order to accomplish these goals, we came up with the following experimental design. We installed a number of miniature recirculating gravel filters that contained various planting densities ranging from 0 to 30 plants per meter squared. These gravel filters were designed in triplicate so that we could draw statistical conclusions about the effects of planting density on nitrogen transformations. All of the effluent from these recirculating gravel filters was combined and subsequently routed into one of two denitrifying media columns, which would provide further nitrogen removal. The wetland themselves were a pretty simple design. They are unsaturated systems filled with pea stone. They were constructed in plastic or wooden frames beneath the surface. The frames were divided into 4 equal sections by placing wooden partitions to create miniature wetlands with the dimensions of around 1 meter squared. We installed 2 sampling ports in each of the miniature wetlands so that we can get a vertical sampling resolution. The system was designed to receive 220 gallons per day, which for each wetland is around 2 gallons per square foot per day. So this loading rate is higher than single pass filters, but it's not the highest loading rate possible for recirculating systems. The systems were planted with Typha latifolia, or common cattail, which is a native plant species in the Northeast. And this species has been shown to have enhanced nutrient removal capabilities, so that's why we selected it for this study. The picture on the top shows the time of initial planting. You can see 2-inch plugs of cattails were planted in the wetlands. The white pipes going across the screen represent the placement of the distribution lines. At the time that this picture was taken, the distribution lines had not yet been installed. They were installed and subsequently covered with some soil so that they were not exposed to the atmosphere. These were subsurface distribution systems. The picture on the bottom represents the same system just several months later after the initial planting, once the systems had started to receive flow. And you can see just by this image that the plants are definitely taking up a lot of nutrients from the system. Okay, so while the main goal of these systems, the gravel filters at least, was to nitrify, and they did do that, we were also pleasantly surprised to find that all of the systems removed significant amounts of the incoming nitrogen. So we found that between 40 and 50% of the incoming nitrogen was removed in all of the gravel filters. And we found that there was no difference in the total nitrogen removal across the different planting densities shown in this figure. However, while we didn't see differences in the total nitrogen removal, we did observe differences in the nitrogen speciation in planted and unplanted systems. This slide shows nitrogen concentrations in planted systems, which are represented by the dashed lines, and unplanted systems, which are represented by the black lines. Ammonium concentrations are on the upper panel, and nitrate concentrations are in the lower panel. And what we'll notice in this figure is that during the first few months of operation, the planted and unplanted systems performed equivalently. However, after this initial establishment period, some pretty substantial excursions in the nitrogen species in planted and unplanted systems were observed. And generally speaking, we saw that unplanted systems exhibited higher ammonium concentrations and lower corresponding nitrate concentrations compared to the planted systems. So if you'll recall, nitrification involves the oxidation of ammonium to nitrate. So if nitrification is happening, we would expect a reduction in the concentrations of ammonium and an increase in the concentrations of nitrate. This is another way of showing what I tried to show in the previous slide. Since the total nitrogen concentrations in the effluent of the different planting densities was not very different, by examining the relative proportion of the various nitrogen species, we can potentially gain some insight into the dominant nitrogen transformation processes occurring. And so in the study, we observed that there were trends with increasing planting density. We found relatively higher proportions of nitrate and lower, which are shown in the white bar, and lower proportions of ammonium and organic nitrogen in the black and gray bars respectively, with higher planting densities. So put kind of simply, we believe that this shows that the higher planting densities have a better potential nitrification, or they're more effective at nitrifying. So in addition to differences in the nitrogen species, planted and unplanted systems can also differ in terms of the ultimate sinks of nitrogen. So in this study, we found that plant uptake constituted a potentially significant nitrogen removal sink. We found between 30 and 80% of the total nitrogen removed was taken up by plants and stored in the plant biomass. It's important to note that this constitutes a temporary pool of for nitrogen because it can get re-released seasonally when plants die. And so if we wanted to permanently remove this nitrogen from the system, management strategies such as harvesting the plants at the time of peak biomass would be critical. So now we've shown you that the wetland systems performed as we wanted them to. They nitrified, and we've shown that nitrification seems to be improved at higher planting densities. But what about the denitrification component? In order to assess that, we're going to look at the entire two-stage system performance over time. The top line in the top figure shows the average influent concentrations over a 15-month period. The middle line, the dashed line, represents the average wetland gravel filter effluent concentration over the same period. And what we see here is that indeed the wetlands did remove about half of the incoming nitrogen. What's shown in the bottom is the average effluent from the two denitrifying media columns. And we can see that these provided substantial nitrate reduction, such that we achieved our target effluent concentrations of less than 10 milligrams per liter. We did a follow-up laboratory experiment where we collected some of the media from the carbon denitrifying media barrels, and we dosed them with synthetic nitrogen-rich wastewater in the form of nitrate to see whether the two different types of media that we used differ in terms of their nitrogen removal capabilities. And while we saw that the wood chips did a slightly better job at removing nitrogen compared to the mulch material, there was a lot of variability between columns, replicate columns. We attribute that to potentially differences in the way that we pack the columns, which could also affect the residence time. Okay, so now that I've showed you a bit about the application of wetlands for nitrogen removal, we want to try and optimize the design of these systems. And that requires us to kind of better understand the critical components in treatment, and as well to better understand the ultimate fate of nitrogen in these systems. The prior systems that I spoke about were operated in unsaturated configurations. And while we still observed a significant plant effect on nitrogen transformations, this environment may not highlight the importance of plant processes. So to better understand this and to determine the fate of nitrogen, we launched a series of laboratory bench scale systems. This is a schematic of the experimental design. We created miniature planted and unplanted wetland systems. They were filled with sandy media, and they were kept saturated. All of the systems received synthetic wastewater at typical loading rates and typical dosing patterns. One very unique aspect of this study was that we applied the use of chemical imaging techniques, which allowed us to take pictures of the oxygen distributions in the soil. So what I'm showing you on this figure, highlighted in these black dashed boxes, are typical images of the oxygen pictures that we would get from this experiment. The color bar on the right-hand side over here represents the concentration. So you can see the red and white colors represent very high oxygen concentrations, whereas the black and purple colors represent low or no oxygen concentrations. And then the colors in between represent a gradient of the oxygen concentrations within those boundaries. Generally, we found that planted systems, shown here, exhibited really heterogeneous patterns of oxygen that were very closely tied to the position of the plant roots. So for example, in this picture, you can almost see the outline of the roots themselves. Comparatively, unplanted systems were characterized by mostly anoxic conditions. The extent of the oxic area or oxygen-rich area was limited to just the upper few centimeters. And so consequently, we believe that this will inherently affect the dominant nitrogen transformation processes. So we hypothesized that the contribution of oxygen from the plant roots and the carbon-rich exudates from plants will promote the processes of nitrification and denitrification in these systems to produce effluent with lower nitrogen concentrations and more N2, which is the end product of denitrification. Comparatively, we expected the unplanted systems to be characterized by very limited nitrification since there were low oxygen conditions. And we expected this limited nitrification to limit denitrification as well, since there was no nitrate available. And so we would expect the effluent from these unplanted systems to have higher concentrations and particularly be almost entirely in the form of ammonium. This figure shows the effluent concentrations in unplanted systems and planted systems, as well as the average influent concentrations over the duration of the experiment. The white bars are, represent the ammonium concentrations, the black bars represent the nitrate concentrations. And what you'll see here is that the planted system did remove more nitrogen than the unplanted system. and that it did a better job at removing the added nitrate to the system. So most of the nitrate that was present in the system disappeared. So in addition to this, we wanted to connect the patterns of oxygen distribution in planted systems to the nitrogen processes. So this figure shows several things. In the black solid line, we see oxygen concentration as a function of distance to a root. We also are showing here nitrate removal as a function of distance to a root, which are just shown in these circles. The negative numbers represent nitrate that was present in excess to what had been added in the influent. So this means that there was some production of nitrate in the system if you have a negative nitrate removal. And what we observed for oxygen was generally at the root, we had lower oxygen concentrations because oxygen is consumed by microbes here. At a kind of very small distance from the root, we have higher oxygen concentrations because the plants release oxygen and it can extend to create kind of a wide oxic region. As we move away from the root, oxygen quickly drops off and gets depleted. And we see how these oxygen patterns correspond to the nitrogen species. At the root, we see some removal of nitrate, which we attribute to plant uptake. At intermediate distances from a root, we see negative nitrate removal values, which again indicate that nitrate is present in higher concentrations than what we added, which suggests nitrification is occurring here. And then again, with increasing distance from a root, when you move away from the oxygen-rich region, we see enhanced nitrate removal efficiencies, almost reaching 100%. And this we attribute to denitrification of the nitrate produced. So in addition to these kind of studies, we wanted to determine the fate of the reduced nitrate in these systems. And we did that by applying stable isotopes. And so just to give you guys a little bit of background information, nitrogen exists in two main forms in the environment, 15N and 14N. The majority of nitrogen is present in the form of 14N or light nitrogen. These are isotopes of nitrogen, which mean that they have similar chemical properties and they differ in terms of their weights. And so this is important because we can apply mass spectrometry to quantify these different species based on their weights. And so in nature, depending on how these forms of nitrogen pair, we can end up with nitrogen gas that has a signal of 28, 29, or 30 N2. In the natural environment, since most of the nitrogen is in the form of 14, we would expect 28 N2 to be the primary species of nitrogen. So we did a number of studies where we actually introduced 15 labeled nitrate into our systems. And so unlike in nature where we would expect a lot of 28 N2, when we introduce 15 nitrogen in excess, we would expect, if it was being denitrified, that we would end up with 30 N2. And so if denitrification was occurring in these systems, we would see a production of 30 N2 over time. And this is in fact what we observed in these systems. So this figure shows the nitrate, the 30 N2 concentrations gathered over an experiment that lasted for a period of 6 hours. And we see a linear increase in the amount of N2 being produced. We also see a corresponding decrease in the nitrate, which is represented by these gray square boxes. And so what we think is happening is this nitrate is being consumed and it's producing this 30 N2 that we observe. So this is a direct evidence that this nitrate that we added is being denitrified. When we compared the denitrification rates in planted and unplanted systems, we noticed that the planted systems had significantly higher denitrification rates compared to the unplanted systems. And by doing some calculations, we determined that the amount of time required to get rid of all of the nitrate added in planted systems would be about 1 day. Whereas in unplanted systems, it would take over 30 days to totally remove all of the nitrate added. Since for these miniature lab systems, the residence time was around 2 days, this means that only the planted systems had the potential to reduce all of the nitrate. So, we've shown in our unplanted systems that we have prevailing low oxygen concentrations that we gathered from chemical imaging. However, when we take effluent measurements in our unplanted systems, we do see that N2 gas is being produced. But we just showed you that there are pretty low denitrification rates in these unplanted systems. And so we started to question whether there might be alternative N2 producing pathways. One such pathway is called anaerobic ammonium oxidation, or ANAMOX. This occurs in oxygen depleted conditions and in the absence of a source of carbon. And so these are exactly the conditions that we would expect in our unplanted systems. In the process of ANAMOX, ammonium and nitrite combine and form N2 gas. So we did another set of isotope work where instead of adding 15 labeled nitrate, we added 15 labeled ammonium to see if N2 was produced. And in fact, we did find that N2 was produced with the 15 ammonium addition, and that it was produced at much higher rates than when we had added the 15 nitrate. So this represents that in this particular case, in unplanted systems, denitrification played a limited role in these unplanted systems, but perhaps other N2 producing pathways were more important here. So just some main takeaways from the research that I've showed you today. Constructed wetlands are capable of achieving our target effluent concentrations, and so they can be applied for wastewater remediation purposes. We found that carbon addition does significantly improve total nitrogen removal, and it allows us to really hit those 10 milligram per liter targets that we're aiming for. We showed you the direct influence of plants on nitrogen transformation pathways, meaning that in these systems, we believe that plants are a really critical component. We also showed that while denitrification is a main nitrogen reducing pathway, in certain systems, under certain conditions, alternative pathways can prevail, such as ANAMOX. Generally, constructed wetlands have been kind of viewed as a black box where we know what's coming into the system and we know what's going out of the system, but we don't really understand what's going on inside. And so the research that I've conducted with the center really aims to kind of clarify these internal processes and better understand the fate of nitrogen in constructed wetlands so that we can design wetlands and optimize the design of these systems to achieve our target effluent standards. So I just want to take an opportunity to thank everyone who's contributed to this work, including our funding source in Suffolk County. and then various representatives from the center. And with that, I'll take any questions. Okay, does anyone have any questions?
Great presentation, Samantha. Have you had a chance? I mean, it's a vast amount of work that you've been able to accomplish. I'm wondering if you've had a chance to think about what plants you might be studying or want to study next. What are your favorite plants from the point of view of the root system creating oxic and oxic boundaries, which would enhance nitrate removal?
Yeah, that's absolutely correct. And so in particular, the wetland that is installed at the Nature Conservancy really kind of examines different plant species. In particular, we wanted to see whether, your typical wetland plants vary in comparison with kind of more ornamental species. So just from a kind of practical—
Samantha, did you hear my last question?
Can you hear me? Are you able to hear me?
Yes, loud and clear.
Okay. Stuart, are you having difficulties over there?
He had a little issue earlier today, so let's just assume that maybe that's the issue. So why don't you go ahead and continue your answer?
Okay. So as I was saying, at the Nature Conservancy, we're trying to examine different plant assemblages to determine whether differences in nitrogen removal occur. But since wetland plants have adapted to really effectively transport oxygen to the root zone, we do think that those would be the more effective plant species.
Great, thanks.
Samantha, I was going to ask about the N15 data, and
yeah.
You didn't mention. I was just curious. Did you get like full quantitative conversion of the nitrate to the 30N or the N15, or was there some loss or gain, or just curious about that?
Absolutely. So, while we do want to achieve a complete budget and recover all of the nitrogen, we were not able to in this study. And I do believe that that's because in this case, these were systems that were open to the atmosphere. And so the plants can still facilitate the transport of the N2 produced to the atmosphere. And so we were not able to recover all of the nitrogen removed as N2 gas.
Not surprising necessarily,
yeah.
But thank you.
Sure. Are there any other questions?
Sam, there's one in the chat, or a couple in the chat. Do you want them read to you, or do you want to read them out to everyone? And then well, it looks like you're going to be getting them that way. So
okay, yeah, I have a bunch of them.
You can read them out and then think about it and answer, or someone else could read them to you.
Sure, I can see them here, so I can read them.
Okay.
Do you imagine wetlands being part of a waste treatment facilities? So I don't totally understand that question. Wetlands, here we're studying wetlands in their application as on-site technologies, but there have been several studies where larger wetlands have been installed to serve, for example, prisons or larger facilities. And so it's not going to be a centralized wastewater treatment facility but they can be used in greater applications besides just single family households. Don't know if that answers that question. Do you assume that, you're welcome, do you assume that some was lost as N2O? I didn't show the data, but in the MASSTC constructed wetland, we actually did measure the nitrous oxide losses from the system. And we did find that nitrous oxide was produced, but it represented less than 1% of the nitrogen lost. So the species was not a major species produced. Could you comment on the effect of leaving biomass in place on leaching of total nitrogen for the planted systems? Sure. So I do, in the case of wetland plants, some of the nitrogen does get translocated in the winter months between the above ground biomass and the roots, but some of it also does get lost to the system. And so there is, and we did observe kind of an increase in, for example, ammonium concentrations in the winter months from planted systems. Can you remind me what organic nitrogen is? So it's basically, in this case, it can be, you're welcome. Let me see if I have an image of it. Yeah, so the organic nitrogen is just sorry, too many questions coming in, is just a natural component of our wastewater, but it also, it's produced in natural processes, but in certain nitrogen transformation processes, organic nitrogen can get converted to ammonium. And so we really believe that that's the main form of the nitrogen in these systems. So most of the organic nitrogen does get converted. Did you measure DOC at the effluent? We did. I didn't show it here as well. The effluent DOC concentrations in in all of the systems was about the same. It was between 12 and maybe 20 milligrams per liter. And so we believe that this carbon that's left in the effluent represents a recalcitrant form of carbon, meaning that it's not really easily utilized by the microbes. And so, for example, plants provide a much more labile source of carbon in the form of low molecular weight organic compounds that are easier to use. I'm not sure if that answered the question. All right, well, thank you everyone for taking the time to listen to me present today. I just want to let everyone know that we have this monthly seminar series with the center, and I believe next month's seminar is going to be provided by Frank Russo and Sue Wei on our next generation NRBs for wastewater treatment. Thank you, everyone.